Antibodies and Disease. So this lecture is going to be divided into 3 parts. In the first part we're going to look at the application of antibodies and why they're useful. In the second part, the genetics and structure of antibodies. And in the third part, how we make antibody-based drugs and where our research fits into that. Part 1. Applications of antibodies. So, starting off looking at the background and the natural role of antibodies, as you'll appreciate, antibodies are part of the adaptive immune system, and this is an incredibly complex system. However, in this single slide, I try to summarize the main role of antibodies in that. So we have B cells. These are antibody-producing cells which express surface IgM and IgD antibodies, and these are membrane-bound. And by chance, one of those happens to interact with an antigen. Now, through a complex procedure which includes expression of the antigen as fragments on MHC, interaction with T-cell receptors, and so on, we get amplification of these B cells that happen to bind to this antigen. So this is the clonal expansion. We also get a process of maturation of the antibodies. So through somatic hypermutation, we get extra mutations being introduced into the antibody sequence. And by chance, some of these antibodies will bind better to the antigen and some of them will bind worse. Now, the ones that bind better will result in this amplification increasing. The ones that bind worse will not be amplified. So we have a mini evolutionary process going on, leading to optimization of the binding for the antigen. And this can then also lead on to so-called class switching, where instead of producing IgM and IgD antibodies, we go on to produce other antibodies that are secreted from the cell, including IgG, which is the major plasma type of antibody. This then binds to antigen, it blocks it, stopping it from being able to interact with cells. It undergoes a process known as opsonization, where this coating of, for example, a bacterium with antibodies leads to things like macrophages eating up the antigen, and it can trigger the complement system, which causes a membrane attack complex to punch holes in the bacterial membrane. So what do antibodies look like? We'll come back to look at the structure in more detail later, but these are some simple kind of overview diagrams. It's a Y-shaped molecule with 2 identical arms. So this is a ribbon view, this is a space-filling view. Down here in orange we have sugars that are involved in the interactions between these parts of the structure. On one arm we have a light chain in green and a heavy chain in blue, which comes right down to the bottom, and we have the same in the other arm, a light chain in green and a heavy chain which comes all the way down to the bottom. And this is another view showing how the antibody interacts with an antigen on the surface of a cell here. So this arm is the bit that undergoes the interactions with the invading cell. So why are we interested in antibodies? What are they useful for? Well, they have all sorts of different types of applications. To start with, they can be used in vitro. We can use them in purification of, for example, proteins. Basically what we do is anchor an antibody onto beads in a column. We put our mixture down the column, and the thing of interest sticks to the antibodies. We can then wash off anything that's an impurity, and finally elute the protein of interest from the antibodies that are on the beads. We can use antibodies in staining, so we can have an antibody that binds to a particular cell component, and we attach something like green fluorescent protein to that, and there are modifications to GFP that cause it to fluoresce in different colours like blue and red, and so we can have these coloured images which show us where particular molecules are found. We can use antibodies in ELISAs, so this is showing a sandwich ELISA where we have antibodies fixed on a plate and another indicator antibody binding to another part of an antigen. So this is the sort of thing that's used in lateral flow tests like pregnancy test kits, and the lateral flow tests being used for COVID. And this is, of course, an example of a pregnancy test kit. Now, Paul Ehrlich, who was a German Nobel Prize winner in 1908, born in 1854, first suggested the idea of magic bullet drugs. In other words, drugs that would target a particular molecule, would go to the site of that molecule without having side effects throughout the rest of the body. And really, antibodies fulfill that kind of desire. So in vivo, they can be used for all sorts of things, just as they can be used in vitro for different things. They can be used in imaging, so gamma imaging, PET scans, guiding surgery. Here's an example here, this radio-guided surgery where a radio-labelled antibody is administered. It's taken up by the body and focuses itself in particular places. For example, in colorectal cancer, you might use an antibody against CEA, that's carcinoembryonic antigen, which is a marker for colorectal cancer. So once you've done that, you can use essentially a Geiger counter to detect where you see a focus of radiation, and you can then excise that small metastatic cancer. In practice, these sorts of techniques are used less now because scanning methods have improved so much. Things like MRI scans and CT scans have got a lot better. Secondly, we can use antibodies for targeting. So that can be direct targeting, drug conjugates, it can be targeting of radioactivity, or it can be indirect targeting using a technique known as ADEPT. So how do we do this? Well, with the direct targeting, we have an antibody that binds to an antigen of interest, again usually on a cancer cell, and we link some sort of cytotoxic agent that will kill cells to that antibody. Now, the linker is designed such that the cytotoxic agent will be released when the antibody is taken up by a cell. So we can do that with drugs, we can also do it with radioactive elements, and there are a number of drugs that have been designed in this sort of way, and a particularly interesting one is this trastuzumab emtansine. Trastuzumab, also known as Herceptin, is an antibody that's widely used in HER2 positive breast cancer. This trastuzumab emtansine is an ADC which has all the normal functionality of trastuzumab, but also has this emtansine drug attached to it, which gives it a second killing mechanism. The antibody-directed enzyme prodrug therapy, or ADEPT, is an interesting idea which unfortunately hasn't really worked terribly effectively. The idea here is that we have an antibody which binds to a tumour cell and has an enzyme conjugated to it. Now you inject that and it localizes at the tumour cell. You now inject a prodrug, which is an inactive precursor of a drug, which can be activated by the enzyme. And this means that you get then the drug being concentrated at the tumour cells and some sort of leaving group here which dissociates from the drug. Now, this is all very well in principle, but the concentration effect that you get from doing this seems to be rather small, and therefore you need to have highly toxic drugs which tend then to leak away from the tumour site and cause toxicity elsewhere. So there is not a huge advantage to doing this over conventional chemotherapy. However, it has been tried out, and this is an example where doxorubicin prodrug is activated by an antibody called 38C2. So in this particular case, not only do we have a tumour-specific antibody, but that's coupled to a catalytic antibody. And these are fairly unusual antibodies that can be created in the lab to catalyze specific reactions. The third category then is therapeutic antibodies, and that to me at least is the most interesting application. These fall into three broad categories. The first is immune activation, where we're triggering the immune system. The second is direct effects of binding, in other words, really working like conventional drugs. And the third is coupling, and we'll see an example of that in just a moment. There are lots and lots of antibodies now that are used as drugs. So here's just a small range, including a number of anti-cancer drugs and a number used for all sorts of other applications, ranging from recovery from angioplasty through rheumatoid arthritis, asthma, multiple sclerosis, high cholesterol, psoriasis, lupus, macular degeneration, prevention of transplant rejection, and viral infections. There are endless different applications these days. So as of the 18th of January 2021, there are 8 antibodies that have been on the market and now withdrawn, generally for marketing reasons, because other drugs have come along that are more effective and or cheaper. There are 94 currently approved in the US or Europe, 16 in review in the US or Europe, which undoubtedly will be approved fairly soon, and 13 approved elsewhere. With the inevitable interest in COVID, there are currently 217 different antibody discovery programs for 62 different targets. The majority of these being antibodies that target the spike protein on the surface of SARS-CoV-2. And of those, there are at least 3 with emergency use authorization. Casirivimab and imdevimab, also known as the Regeneron cocktail, produced by a company called Regeneron, are the 2 antibodies that President Trump was given, and bamlanivimab is another one produced by Eli Lilly. So let's look at the first of those kind of categories, which is triggering the immune system. So this is an example of an antiviral antibody called palivizumab or Synagis, which binds to respiratory syncytial virus, or RSV, and RSV is the primary cause of death in premature babies. So what we're doing here is producing an antibody against this virus, leading the immune system to kill the virus. Of course, the COVID antibodies, this is again the idea here that we're targeting with a lot of these antibodies the spike protein on the virus and causing antibodies to be produced that will either simply block the access of the virus to its receptor or will lead to killing of the virus. There are also many anti-cancer antibodies where the aim is that the antibody binds to a tumour antigen, leading to killing of those cancer cells. The second category then is working as conventional drugs. So how do conventional drugs work? Well, they are generally small organic molecules which bind into tight pockets in proteins. Now that is really a limitation of a small molecule drug. It has to bind into a small pocket because the drug itself is small and therefore can only make a limited number of interactions. Therefore, in order to optimize both the affinity and the specificity, you need to make as many interactions as possible, and you achieve that by binding into a small pocket. Now that means that many targets are not druggable using small molecule drugs. So if you think of wanting to disrupt, for example, a protein-protein interaction, or you want to bind to something having a convex surface, for example, a hormone that would normally bind into a receptor, in those instances, small organic molecules won't work, but antibodies become ideal. And this slide just shows you, for kind of scale, a bit of a comparison. Here we have just part of an antibody, so we saw that Y shape of the antibodies. This is just the ends of those arms, or the end of one of those arms, showing you the approximate size. And this is a phosphocholine molecule. Okay, that's rather smaller than most drug molecules, but a typical drug molecule might be round about this sort of size that I'm indicating here, compared with this being the binding surface of an antibody. So the large surface can therefore interact with a much larger range of molecules, and you get high specificity and high affinity to flat or convex surfaces that you cannot get a small molecule drug to interact with. And the third category then I described as coupling, and here we produce multi-specific antibodies. Now that can be done in a number of ways, and we'll come back to that later on, but one of the simplest ways, at least conceptually, is that you have these two arms on an antibody. So if you make one of the arms bind to one thing, the other arm bind to something else, then you can achieve cross-linking between those two things. So an example here would be to have one arm binding to killer T cells, typically by binding to the surface molecule CD3, and the other arm binding to a cancer cell, so that you target the T cell to the cancer cell. At that point, we're going to take a break and move on to genetics and structure in part 2.